My today's topic of teaching is bioenergetics and glycolysis. Now, in this class, we'll learn how the energy is getting changed inside the cell. Now, when we are talking about any living system, then we have already learned that there are different activities which are going on inside the cell. And those activities, when it is taking place, it is the energy driven process. Now, when we are studying the bioenergetics, it is the study of flow and transformation of energy in and between the living organism and between the living organism and their environment. That means it may be within the cell or it may be the cells and their surroundings. The study of the balance between energy intake in the form of food and energy utilization by the animals for life sustaining processes. Now, when we are talking about the different activities in any living system, obviously we are talking about the food. We are taking food as a source of energy. Now, when we are taking this food, we are taking some of the macromolecules, what I have already talked and I have already told you about this carbohydrate, fats, proteins and so on. Along with these macromolecules, some of the micromolecules are also being assimilated within the living system. Now, when these macromolecules along with these micromolecules are taken, ingested by the body, then it is getting assimilated, it is getting absorbed, it is getting digested, and then it forms the energy, it produces energy. And in any living system, when we are talking about the energy, we mainly talk about the ATP energy. Now, ATP is one of the major important form of energy. Now, ATP is catalyzing different activities, different such reaction, and when these reactions are going on inside any living cell, it produces some amount of heat. Now, this obviously when it is this heat is being produced, the different functions, physiological functions are also simultaneously going on, and we are calling it as the cell is performing some activities or work. Now, when we are talking about the transformation of energy from one form to another, we cannot devoid of any some terminologies which is called as metabolic activities. Now, what is the metabolism? Metabolism is an universal process. It is complex and irreversible process. Now, the entire metabolic activities of any living cell can be of two types. Either it is the anabolic process or it is a catabolic process. Now, what is the anabolic process? When the cell is synthesizing something, it is called anabolism. Now, here the building block of precursors are there within the cell and some synthesis is going on. It is the endergonic process and some product is being synthesized by the system, by the cell. And when it is the catabolic process, the macromolecules which are present within the cell, say in the form of carbohydrate, fats, proteins and so on, these macromolecules are is broken down into micromolecules, that is macromolecules are getting choked into micromolecules, and this way the different functions or different activities are being performed by the cell. Now, photosynthesis is one of the example of the anabolic process, and respiration is another example which is going on inside the cell is the catabolic process. Now, when I'm talking about the different reactions which are simultaneously going on in any living system, it follows the laws of thermodynamics. Now, what are the laws of thermodynamics? That energy cannot be created nor be destroyed, but it can only be transformed from one form of energy to another. That means from potential energy to the kinetic energy. Now, when we also talk about any reactions which are going on inside any living cell, we are talking about the entropy or the disorderness of the system. Entropy is the extent of disorder or the randomness of the system and becomes maximum as equilibrium is approached. Now, this energy often stored as potential in the form of bond. Now, this energy is required to break the bonds and hold the atoms in the molecule together. That means it tries to aggregate. The amount of energy that is available to break such bonds and form such a bond are called the free energy. Now, what is this free energy? It is symbolized as delta G. Now, to determine whether a particular reaction will take place in a cell, we use the concept of free energy. If we see the relationship of this free energy, enthalpy and entropy, then it can be expressed as delta G is equal to delta H minus T delta S, when delta H is the change in enthalpy, T is the temperature and delta S is the change in entropy. Now, when we are talking about the free energy of any reaction, now this free energy change, that is the delta G of any reaction, determines its spontaneous nature, spontaneity of any reaction. A reaction is spontaneous and it is favorable if the delta G value is negative. That means if the free energy of products are less than the reactant, then only we can tell that this delta G is delta G, when it comes as negative, that means it is a spontaneous reaction. That means that reaction is feasible in nature. Now, for any reaction, suppose if we take the reactants as A plus B, which gives rise to C plus D, which are the product, then we can find that delta G of this reaction is delta G0 prime plus RT ln concentration of the product versus by the concentration of reactant. When delta G0 prime is the standard free energy change at pH 7, 1 molar reactants and products, where R is the gas constant and T is the temperature of the reaction. Now, when we are talking about if there is no mention of the temperature, now here we can think of that reaction has taken place for any biological system at 37 degrees Celsius. Now, here delta G0 prime is negative RT ln K equilibrium. Now, how it has come? Now, as we have already mentioned, I have already shown you that delta G is equal to delta G0 prime plus RT ln concentration of product by concentration of reactant. Now, here at equilibrium, we know delta G, or the free energy of any reaction, is 0. Now, if this is so, then we can write this equation as 0 is equal to delta G0 prime plus RT ln and concentration of product by concentration of reactant, or it can be rewritten as delta Z0 prime is equal to minus RT ln concentration of product by concentration of reactant, or K equilibrium is the concentration of product by the concentration of the reactant. So, we can write, and if K prime equilibrium is denoted as concentration of product by concentration of reactant, then we can rewrite that delta Z0 prime is equal to minus RT ln K equilibrium. Now, when we are talking about this particular K equilibrium, it is the exponential of delta G0 prime by RT. So, when we are defining the K equilibrium, that is the concentration of product by the concentration of reactant, delta Z0 is equal to RT ln into K equilibrium. It can be otherwise written as that K equilibrium is the exponential of delta G0 prime by RT. That means K equilibrium is exponentially dependent on the change of this delta G0 prime. Now, when we are talking about this, the different variation of equilibrium constant at delta G0 prime at 25 degrees centigrade. So, here the reaction which is carried out at 25 degrees centigrade, and if we see that K equilibrium and delta G0 prime, that is kilojoule per mole, and if we see the starting of the starting with 1 molar reactant and product in the reaction, then we can find that here if you see that the order of reaction here, this K equilibrium is 10 to the power 4, and here this delta G is only this minus 23 kilojoule per mole. Now, we have to have a very serious notice of this ln, that is the exponential part, and if the change in this, the order of reaction is in the form of 100th order, the change in K equilibrium is merely by the single unit. See here the order of this reaction is 100th, and here there is a minor negligible change in the delta G0 prime, and this is when this delta G0 prime is negative, it indicates the reaction is spontaneous and it proceeds forward direction. At equilibrium, delta Z0 prime is 0, and when it is in the positive, delta Z0 is positive, it indicates that the reverse, it is just reverse of this earlier equation, earlier conditions of the cell, and it is telling that it it is the non-spontaneous reaction. Now, here this is the indication of this K prime equilibrium and delta G for any reaction. Now, as I have already mentioned that if delta G is positive, this means that the product contains more free energy than the reactant, and it do not proceed spontaneously and it needs energy. These reactions are called endergonic reaction. Now, here you see this is the condition. If we are taking this particular reaction condition, here it is the reactant, and reactant is converted to the product. Now, here to carry out this reaction, that energy is supplied. Now, energy is being utilized to break this bond to convert the reactant to the product. Now, here if this product is formed, you see this product has got more energy than this reactant, and when this product has got more energy than this reactant, then it in any point of time it may happen that this reaction may revert back, or it may go for some other intermediate product formation or coupling reactions and so on, and this product is considered to be an unstable product. Whereas in this condition, here also energy is needed to start the reaction, to initiate the reaction, and here we can see that this particular when energy is given to this system, it is converted to this product, and when this product is formed, you see the energy which was required to convert this reactant is much lower form than this initial reaction, and that is the indication that delta G of this particular condition is negative and process is feasible. Now, here this energy is being released, and here in this case it is just reverse. Here the energy is being supplied. Now, when we are talking about this type of reaction, we are mainly talking about the activation energy. Now, what is this activation energy? If all chemical reactions that release free energy tend to occur spontaneously, why have not all such reactions already occurred? Now, the energy required to break the chemical bonds and to initiate the chemical reaction is called the activation energy. Now, activation energies are not a constant, but they can lower the catalysis, the catalytic requirement that stress the particular chemical bond, and these bonds become easy to break. Now, here if we see two such reaction, one is catalyzed by the catalyst and one reaction if we see that without the help of any catalyst, then we can find that say reaction is taking place, and when it is reaching to this peak, it is the activation energy. This is the activation energy which is required to transform this reactant to the product which is stabilized. Now, in another situation, when in presence of catalyst the reaction is taking place, it needs much lower energy. That means the activation energy to reach the much lower energy is required to reach the transition state, and then the product is being produced. And when such type of product formation is there, it is called in presence of the catalyst. and enzyme mediated any such reactions are very spontaneous, and transition state energy or the activation energy is much lower compared to the uncatalyzed reaction. Now, here when we are talking about this energy coupling, a spontaneous reaction may drive a non-spontaneous reaction. Energy changes of coupled reactions are additives. For example, when we are talking about the first stage of glycolytic process. Now, this particular step I will be coming a little later. Now, here I want to tell you hexokinase is the enzyme which is catalyzing this reaction, and in this reaction what is happening? Glucose is getting phosphorylated and it is converted to glucose 6-phosphate. Now, here if we see the reaction is catalyzed in the glycolytic process by the enzyme hexokinase, and if we see the half reactions, then we can find that ATP plus water is giving rise to ADP plus PI, and if we see the delta G prime G0 prime, it is negative and minus 31 kilojoule per mole. Now, when we are talking about the second half, then this inorganic phosphate plus glucose is getting converted to glucose 6-phosphate plus 1 molecule of water, which is the delta G is positive and 14 kilojoule per mole. Now, if we couple this reaction, then we will find that this ATP plus glucose is getting converted to ADP plus glucose 6-phosphate. This is the ultimate product. Now, if we break down the half reaction, then we can find that one is the spontaneous reaction, another is the non-spontaneous, but when we couple these two reactions, we will find that it is the ultimate result is the spontaneity of this particular reaction. Now, two separate reactions occur in the same cellular compartment, and one spontaneous and other is not, may be coupled by a common intermediate, that is the reactant or the product. A hypothetical but typical example involved in pyrophosphate. Now, enzyme 1 is, if we are considering enzyme 1, then it is converting A plus ATP, which is getting converted to B plus AMP plus PPI, that is pyrophosphate, and delta G0 prime is 15 kilojoule per mole. When we are talking about the second part, then we are talking about this PPI, which is the end product of the first, plus water molecule, it is being converted to 2PI. It is the spontaneous reaction. Now, when we are summing up these two reactions, the overall spontaneous reaction, if we sum up, then we can write A plus ATP plus water is resulting B plus AMP plus 2PI, and it is a spontaneous reaction. Now, pyrophosphate is often the product of a reaction that needs a driving force. Its spontaneous hydrolysis catalyzed by the pyrophosphatase enzyme drives the reaction for which the pyrophosphate is a product. Now, obviously, we are talking about high energy bond. Now, phospho anhydride linkages are said to be the high energy bond which is present in the ATP molecule. Now, here bond energy is not high, just delta G of hydrolysis is negative. Now, when we are talking about the high energy bonds, which is represented by this particular symbol, and it represents the high energy phosphate group with a large negative change in the delta G of hydrolysis. Now, here it is the adenine moieties which is attached to this ribose sugar. That first carbon of this ribose sugar is occupied by the adenine molecule, and to the fifth carbon of this ribose sugar, 3 phosphate groups are attached, and they are attached with the phosphoanhydride linkages, and this is the ATP molecule which is there in the cell, and we generally refer that this is the form of the energy which is being utilized by the living system. The compound with high energy bond are said to have high group of transfer potential. For example, inorganic phosphate may be spontaneously cleaved from ATP for transfer to another compound and hydroxyl to a hydroxyl group on glucose. Now, if we see the phosphoanhydride linkage which have a very high delta G of hydrolysis, then we can find that because of this resonance stabilization of the product of hydrolysis which exceeds the resonance stabilization of the compound itself, and also we can find that the electrostatic repulsion between the negatively charged phosphate oxygen atom favor the separation of the phosphate, and that is the reason of the phosphate anhydride linkages which have the very high delta G value. Now, if we take this ATP as the standard, then we can find that it has got low energy phosphates as well as high energy phosphates. Now, when we are talking about the phosphoenol pyruvate, carbamoyl phosphate, 1,3-bisphosphoglycerate to 3-phosphoglycerate, creatine phosphate, etc., these reactions will find that these particular energies which are produced during the reactions are coming under the high phosphate bond, high phosphate energy, and these particular reactions are otherwise known as substrate level phosphorylation, which I'll be coming later. And here the sum of these reactions which are there taking place within the system are the low energy phosphate. So, these are the general categorization of the standard free energy of hydrolysis of some organophosphate compounds. Now, based on this delta G value, we can differentiate this type of category. We can categorize in this fashion. Now, when we are talking about this ATP, in the living cell, the principal energy, high energy intermediate or the carrier compound is adenine triphosphate. Adenine triphosphate has a special role in energy coupling and the inorganic phosphate transfer. ATP often serves as the energy source, and hydrolytic cleavage of one or both the high energy bonds of ATP is coupled to an energy requiring non-spontaneous reaction. That means, we can expect two phosphate group which may be cleaved by the ATP molecule. Now, if we see some reaction that two phosphate group can be cleaved as two phosphates are released by the hydrolysis of ATP, as I have told that AMP which is having two phosphate, that means in other way we can tell ATP, and when ATP is getting dephosphorylation reactions are going on, we can get AMP or having P, or we can tell it that adenine diphosphate and one PI. And when ADP is AMP and linked P is further depleted, it forms AMP plus PI. That means ADP to AMP and PI. Alternatively, AMP and 2-phosphate gives rise to AMP plus 2-phosphate together, which is further converted to 2-inorganic phosphate. That means this pyrophosphate is converted to inorganic phosphate, and PPI is giving rise to 2-PI. Now, AMP functions as an energy sensor and regulator of any metabolic activities. When ATP production does not keep up with the need, a higher portion of the cell's adenine nucleotide pool is the AMP. AMP stimulate the metabolic pathway that produce ATP, and some example of this role which involved direct allosteric activation of the pathway enzyme by the AMP. Some regulatory effects of AMP are mediated by the enzyme, that is AMP activated protein kinase. Now, here, as I have already told you about the ATP, some artificial ATP analogues have been designed that are resistant to cleave the terminal phosphate by hydrolysis. Now, what is that molecule? It is called AMP-PNP. Such analogues have been used to study the dependence of coupled reaction on ATP hydrolysis. In addition, they have made it possible to crystallize an enzyme that catalyzes ATP hydrolysis with an ATP analog at the active site. So, you can see here one nitro group is being attached to this particular moieties to resist the cleavage of the terminal phosphate group of ATP, and this is called the ATP analog. Now, inorganic phosphate in many substances, in many organisms, they store the energy as inorganic polyphosphate. A chain of many phosphate residues are linked together like P, and in this way, and it forms the phosphoanhydride bond. Hydrolysis of PI residues from polyphosphate may be coupled to energy dependent reaction. Depending on the organism or cell type, inorganic polyphosphate may have the additional functions. Say for example, it may serve as the reservoir of PI, a chelator of metal ions, a buffer or a regulator of any reaction. So, if we see the biological system, the living system use the chemical energy to power the living processes. Exergonic reaction takes place simultaneously with the loss of free energy, that means the delta G is negative, and endergonic reaction requires to gain the free energy, that is delta G is positive, and only occur when coupled to exergonic reaction. ATP acts as the energy currency of the cell, transferring the free energy derived from substances of higher energy potential to those of lower energy potential. And this way, the different reactions, the different catalytic reactions are carried out in any living system. Now, here with this I am just concluding this bioenergetics. Now, we will be coming to the actual reaction system in the living cell which is being carried out in some of the metabolic activities. Glycolysis is one of such example where glycolytic pathway describes the oxidation of glucose to pyruvate, pyruvic acid, with the generation of ATP and NADH, and it is also otherwise known as Embden-Meyerhof pathway. Glycolysis is the universal pathway. It is present in all living organisms, starting from yeast to mammals. This is the process which takes place in the cytosol, cytoplasmic fluid of the cell of the prokaryotes and eukaryotes. Here no energy is required for, no oxygen is required for carrying out this particular process. Now, if we see the glycolytic process, the first step of glycolysis is glucose to glucose 6-phosphate. Now, if we see this entire reaction, the entire glycolytic process can be divided into 10 different steps. Now, these 10 different steps can be once again divided into 2 stages. Now, first stage and second stage. So, let us start with the first stage of glycolysis. Now, in the first stage, glucose is getting converted to glucose 6-phosphate. Now, within a cell, glucose 6-phosphate is produced by the phosphorylation of glucose. That means, this is the glucose moieties, and here glucose 6-phosphate means on the 6th carbon atom, here this phosphorylation is taking place. And here when we are talking about this particular reaction, here one molecule of ATP is being utilized to produce this ADP, and this phosphate which is being transferred is coming and phosphorylating this glucose molecule, which is the starting substrate for this reaction. Now, here besides this glucose, there may be some other hexose sugar which can also participate in this reaction. This reaction is catalyzed by the enzyme which is called hexokinase. Now, in this bioenergetics, I told you that hexokinase is one of the example of glycolytic process. Now, here any type of ketohexose or aldohexose can be converted to glucose 6-phosphate. Now, here if we see the affinity of this hexokinase enzyme, then we can find that the affinity of hexokinase is more towards the aldose sugar than the ketose sugar, like mannose, galactose and so on. So, glucose, and they are converting, this enzyme is getting this helping to convert this, it is catalyzing the reaction to convert glucose to glucose 6-phosphate. This is catalyzed by the enzyme hexokinase in most cell. That hexokinase is present in every cell starting from the higher animals to the lower. Similarly, the same reaction can also be carried out with another enzyme which is called the glucokinase. The name itself indicates that glucokinase has got more affinity than the hexokinase enzyme. This glucokinase is present in the liver cell of any animal, and one molecule of ATP is being used in this particular reaction. If we compare the kinetics properties of this hexokinase and this glucokinase enzyme, then we can find that as glucokinase has got only specificity towards glucose, the Km value of glucose is much lower compared to this hexokinase enzyme. Now, this hexokinase enzyme is is present in the liver and it is absent in the muscle. Now, when glucose concentration is high, glucokinase is active, but this glucokinase enzyme is absent in any diabetic patient. Now, this is the first stage, and this both the enzyme hexokinase and glucokinase needs the metal ion like Mg or Mn for catalyzing this reaction. The second step of this glycolytic process is glucose 6-phosphate to fructose 6-phosphate. This is the isomerization reaction. Phosphoglucose isomerase is the enzyme, and they are converting this aldose sugar to the ketose sugar. In this reaction, glucose 6-phosphate isomerized to fructose 6-phosphate by phosphoglucoisomerase. Now, in the third stage, fructose 6-phosphate is getting converted to fructose 1,6-bisphosphate. Now, in this particular reaction, phosphofructokinase is the enzyme which is playing a significant role. This enzyme is so important that it is otherwise known as the rate limiting step of glycolysis. Now, in the cytoplasmic reaction, one molecule each of fructose 6-phosphate and one molecule of ATP is converting the fructose 6-phosphate to fructose 1,6-bisphosphate. Now, one molecule of ADP is produced in this reaction. This reaction is irreversible under physiological condition. Now, what is happening in the first step when glucose is phosphorylated to glucose 6-phosphate, one molecule of ATP was utilized, and see here another molecule of ATP is being utilized for getting this fructose 6-phosphate to fructose 1,6-bisphosphate. That means the first carbon and the sixth carbon are getting phosphorylated with this ATP, and ATP is converted to ADP. Now, if we see the enzyme phosphofructokinase, this particular enzyme, it is the allosteric enzyme. It is a big, huge in structure. 3,80,000 is the molecular weight of this particular enzyme. It can stimulate and modulate the allosteric function, and when in the cell ATP concentration along with the citrate or long chain fatty acid concentration is more, it inhibits the phosphofructokinase, and when this enzyme is getting inhibited, the entire reaction is getting stopped. When in the cell the ATP concentration is much lower, and then this phosphofructokinase is once again getting stimulated, or it is switched on, and it is converting this fructose 6-phosphate to fructose 1,6-bisphosphate. The glycolytic process up to this step is now irreversible, and the energy supplied destabilizes the molecule. Because the reaction catalyzed by the phosphofructokinase is not energetically very favorable, it is essentially irreversible, and the different pathways must be used to do the reverse conversion during gluconeogenesis, and this makes the reaction a key regulatory point of glycolytic state. Now, when fructose 1,6-bisphosphate is produced with the help of this enzyme aldolase, it is being cleaved to two molecule of 3 carbon compound. One is called dihydroxyacetone phosphate, another is the glyceraldehyde 3-phosphate. Now, earlier what we have seen, 1 6-carbon hexose molecule was there. Now, this 1 hexose molecule is now splitted to 2 molecules, 3 carbon each. One is the dihydroxyacetone phosphate, another is the glyceraldehyde 3-phosphate. Aldol split the hexose ring of 1,6-bisphosphate into 2 triose sugar, that is the dihydroxyacetone phosphate, and glyceraldehyde, that is the aldose sugar. Now, glyceraldehyde 3-phosphate is in the presence of triose phosphate isomerase enzyme can be once again getting converted to each other. That means isomerization of ketose to aldose sugar is taking place very fast within the cell. The enzyme triose phosphate isomerase rapidly interconvert the molecules of dihydroxyacetone phosphate and glyceraldehyde phosphate. Glyceraldehyde phosphate is removed as soon as it is formed to be used in the next step of glycolysis. That means as soon as dihydroxyacetone phosphate, this ketose sugar is converted to aldose sugar, this glyceraldehyde 3-phosphate is immediately being taken up in the next stage of glycolytic process. Now, if we see this glyceraldehyde 3-phosphate, this glyceraldehyde 3-phosphate in the presence of dehydrogenase enzyme, it is once again getting phosphorylated to 1,3-bisphosphoglycerate. Now, the enzyme glyceraldehyde 3-phosphate dehydrogenase serves two functions in this particular step. The first of the enzyme transfers the hydrogen from the glyceraldehyde phosphate to the oxidizing agent of nicotinamide adenine dinucleotide. That means here this NAD is being converted to NADH. Next, glyceraldehyde 3-phosphate dehydrogenase add a phosphate from the cytosol to the oxidized glyceraldehyde phosphate to form 1,3-bisphosphoglyceric acid. This occurs for both the molecules. Now, what is happening? Earlier we were talking about 1 hexose, now we are talking about 2 triose sugar. That means these triose sugars are 2 molecules, that is 1 hexose is converted to 2 molecules. Now, when 1,3-bisphosphoglycerate is produced, that means 2 molecules of such compounds were produced. So, this occurs for both molecules of glyceraldehyde phosphate to produce in the next step. Now, henceforth, whatever may be the steps were 2 molecules of each tricarbon molecules. So, 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate with the presence of the enzyme phosphoglycerate kinase. The enzyme phosphoglycerate kinase transferred the phosphate from 1,3-bisphosphoglyceric acid to a molecule of ADP to form the ATP, and which is called as substrate level phosphorylation. That means it is losing one phosphate from this core moieties, and ADP is being taken up and it is produced the ATP molecule. And as 2 molecule of this 1,3-bisphosphoglycerate is there, so 2 molecule of ATP is being produced in this particular step. This particular step is otherwise known as the substrate level phosphorylation. The substrate level phosphorylation is a type of chemical reaction that results in the formation of creation of ATP by the direct transfer and donation of phosphorylation phosphate 2-adenine diphosphate from the reaction intermediate, and conventionally, however, the phosphorylation group that is transferred is referred to as phosphate group, and substrate level phosphorylation occurs in the cytoplasm of the cell as a part of the glycolytic process and the mitochondria in mitochondria for the Krebs cycle under the aerobic and anaerobic process. Now, when we are talking about this 3-bisphosphoglycerate, it is once again the transfer of phosphate group is taking place in the presence of the enzyme mutase, and it is converted to 2-phosphoglycerate. So, this third from the third carbon, it is transferred to the second carbon. Now, when this 2-phosphoglycerate is produced with the help of this mutase enzyme, the next step with the help of enolase, which is releasing one molecule of water from this reaction, and it is converting to phosphoenol pyruvate. Now, this phosphoenol pyruvate happens for each molecule of 2-phosphoglyceric acid. Now, this phosphoenol pyruvate, when it converted to pyruvate in the presence of pyruvate kinase, here also the phosphate group is being utilized by this ADP, and 2 molecule of ATP is being utilized, being produced, and it is another example of the substrate level phosphorylation. And this is one of the this example of this substrate level phosphorylation in the glycolytic pathway. Now, pyruvate, as soon as it is produced, pyruvate is considered to be the last end product of this particular cycle. Now, if we see the overall reaction, then we can find that first in this step, glucose is converted to glucose 6-phosphate, one molecule of ATP is utilized, glucose 6-phosphate to glucose 1,6-diphosphate, here also one ATP molecule is utilized, but here in glyceraldehyde 3-phosphate, you see 2 molecule of NAD is produced, NADH, and 1,3-diphosphoglycerate is produced, that 2 molecules of ATP, and here also 2 molecules of ATP is produced as a result of this reaction. So, if we see the overall energy balance in this particular reaction, then we can find that this is the utilization of 1 ATP and up to glucose 6-phosphate, fructose 6-phosphate to fructose 1,6-diphosphate, and in this henceforth reaction, glyceraldehyde 3-phosphoglycerate to 1,3-bisphosphoglycerate, 2 molecule of NADH, here 1,3-bisphosphoglycerate to 3-phosphoglycerate, here also 2 molecule of ATP, and phosphoenol pyruvate to pyruvate, here also 2 molecule of ATP is produced. Now, as we know that 1 molecule of NADH is giving 3 molecules of ATP, so we can say that it is approximately the 6 ATP molecules, and here 2 ATP molecules, 2 are getting cancelled. So, net ATP molecule gain in this process is 8. Now, once this pyruvate is formed, this pyruvate is being utilized by very many reactions. It can be converted to lactate when there is a scarcity of oxygen in the muscle or in the cell. Under anaerobic condition, it can also produce ethanol, and under aerobic condition, pyruvate is getting converted to acetyl-CoA, and this acetyl-CoA is entering to the Krebs cycle, and what I will be discussing in the next class. And I think today I have tried to give you the overall bioenergetics, the energy utilization process in the living system, and how the metabolic reactions are taking place within the living system. And with this today I am concluding. Thank you very much.